All right, pre-AP biology. Excuse me. Again, we are continuing with our Unit 4 notes on cell reproduction. This lecture today is a big one. It's probably one of the more important ones that I give during the first semester. Not that the other ones aren't important, but this one has a lot of details, a lot of vocabulary. If you can print out the fill-in notes, it might be a good idea for you to to fill those in as I go along. Um, Again, if you need access to those, you can find them in my Buzz folder or on the agenda. Or if you want me to email them to you, I can email them to you or pick them up here at school if you need to. Okay, so what we're going to talk about today is something called the cell cycle. Now, the cell cycle is kind of a fancy word for the lifespan of a cell. Different cells have different lifespans. Some cells live, um, kind of long periods of time, whereas other cells don't live very long. Like your skin cells, for example, they don't live very long. They might live a couple of days and then just die, and your body replaces them fairly quickly. Whereas other cells tend to live much longer. So, again, the cell cycle, it kind of varies from cell to cell, but every cell goes through a series of stages in its life. And this is what we call the cells. I don't care if it's a blood cell or a bone cell or a skin cell, they're all going to kind of go through the same series of steps. Just the length of time it takes to get through those steps kind of depends on the actual cell. But anyway, so what we're going to do today is we're going to talk about the three major stages of a cell's life. And you may have heard of some of these before: interphase, mitosis, which I'm sure you've heard of before, and a final step or a final stage called cytokinesis. So we're going to talk about these things in order. Now, your job, kind of the learning objective, is that you need to be able to describe these stages in detail. All right. I mean, how do we know that I know what I'm talking about? Because I can describe it to you. All right. You need to be able to describe it as well. And if you can, then you probably understand it. So you're going to have to use terms and understand vocabulary in each step of the cycle to really be able to do this. Okay. All right. So this is a picture of the cell cycle. And it looks kind of daunting at first. There's a lot of stuff going on, lots of arrows, lots of colors, lots of vocab, lots of words. But I'm going to kind of go through this with you today, step by step. And again, at any point, if you want to pause the video, if you're kind of getting information overload, then by all means do that. You can go back, rewatch a part again if you need to make sure you grasp it. But what I want us to first understand about this picture is there is a cycle to it, right? I mean, you see these arrows that are just kind of going around and around and around, right? A cycle is basically a series of steps that repeats itself. The days of the week, right? They're a cycle. They repeat themselves over and over. The months of the year, they're a cycle. They repeat themselves over and over again. Well, the steps of the cell cycle are the same thing, right? It's a series of steps and eventually will repeat itself. So what we need to figure out first is, where are we starting, right? What is the starting point of this cycle? Well, we're going to start with interphase. Okay, that's kind of the starting point of the cell. So I want you to take a look at this cell right here. Okay, and this is kind of the starting cell that we're going to be working with today. Now, this cell, it looks kind of simplistic. It doesn't look like there's much to it. But what I want us to understand about it is, let's just assume that this is just a normal somatic body cell. It could be a skin cell or a liver cell or whatever. But we know from previous lectures that somatic cells have 46 chromosomes, right? 23 pairs. So what you're seeing inside of the nucleus, you're not seeing all 46 pairs, or I should say all 23 pairs. You're just seeing two little chromosomes here, okay? So, we can just kind of make an assumption that there's 46 in there because there is. All right, but we're just seeing, I guess, for simplicity reasons, just two little chromosomes. Okay. So we're going to start here, okay, with our regular somatic cell, and we're simply going to follow these arrows as we go through, and we'll talk about each one of these steps again as I get through the lecture today. All right, so we're going to talk about interphase. Now, interphase, is kind of the first three steps, and you'll notice here that if you kind of think of this as like a big pie or something, these particular steps take up a big chunk of the pie. So what that tells us is that most of the lifespan of a cell, probably more than 75% of the time of a life of a cell is spent in interphase. Now, interphase consists of three separate steps. We call it G1, S, and G2. So what we're going to do is we're going to start here, right? This is our starting cell, and we're just going to go through each one of these steps of interphase to kind of understand what's happening to the cell in every single step. All right, so G1, right? This is the first stage of interphase that the cell enters. Now, to kind of remember this is kind of how I remember it. All right, the G stands for growth. So during this first stage of a cell's existence, really, that cell is going to be spending some of its time just getting bigger. So you can probably imagine at this time that the cell is taking in lots of nutrients, taking in lots of energy, and the cell is getting larger and larger and larger. Now, also during this time, this is kind of an important thing. A lot of the organelles, things like mitochondria and ribosomes, right? All those organelles we talked about. Those organelles are getting copied. So what I kind of want us to understand is, at the end of G1, the cell is going to be larger and it's going to have all of its organelles copied. Okay. So we have twice as many ribosomes, twice as many mitochondria, on and on and on. Now you'll also notice at the end of G1, we still have 46 chromosomes. It's still the same number of chromosomes that we've had, two number ones, two number twos, two number threes, two number sevens, okay, whatever, right? Two sex chromosomes, either XX or XY. Now, one thing about the G1 phase is you'll notice here that there's like a little, I guess, fork in the road, I suppose, that a cell could take. And some cells do this, but most of them don't. Right? It says here that some cells leave G1 and they enter what we call G0. Now, if a cell decides to leave G1, it's not going to come back and complete the cycle. So it kind of goes off on its own. All right. It says here, cells that do this never re-enter the cycle and they won't ever divide. So a really good example of this is brain cells. This is why, like, for example, like if you get a brain injury, like those cells aren't going to grow back. Or if you get like a spinal cord injury where your spinal cord cells are damaged, those cells aren't going to grow back again. And that's because those cells have already entered G0. That means they're kind of deciding that they're not going to go through a cell cycle So if they're damaged in any way, then they can't be duplicated. So it's like a really important thing that we don't injure our brain or injure our spinal cord. I mean, we can skin our elbow, right? And those cells will regrow again. But some cells don't do that, or they grow back very slowly. So in that case, they enter that G0 phase. So that's kind of an important thing to remember. All right, so now we're going to enter the second phase. So we've gone through G1. Now we're going to go into the S phase. If you kind of look at the diagram here, you'll probably understand what the big thing is about the S phase. All right, S stands for synthesis. This is the synthesis phase. And what that means is that all the DNA gets copied. Okay, so we've got 46 chromosomes, right, at the end of G1, two number ones, two number twos, two number sevens, etc. But if all of that gets copied, we're going to have 92. Now we have a special little vocab thing here that I think you need to be aware of. So, like for example, your chromosomes, right? Those are kind of these individual units, right? This is a chromosome here, okay? But when it gets copied, we don't call it a chromosome anymore. All right? We call these things chromatids. All right, so we go from 46 chromosomes, in this cell down here at the end of G1, to having 92 chromatids, okay, if that makes any sense. So there's a little bit of a vocab change when we go from G1 to S, but we're just doubling the amount of DNA, doubling the amount of genetic information. Now, there's another vocab word here as well, and this will come into play in a little while. It's called a centromere. A centromere, just kind of think of it as kind of like the middle of the chromosome. But these two chromatids are kind of held together at that area. You kind of think of it as like a rubber band that's like tied around two sticks. Okay, that rubber band is kind of holding those two sticks together. Okay, that's our centromere. Okay, for right now, that's all you really need to know about it. All right, now we're going to move on to the G2 phase. Now again, the G, so we get more growth, so the cell's going to get bigger again. But one of the big things that happens during G2 is the cell kind of double checks to make sure all of those chromosomes were copied correctly. You got to remember about chromosomes, right? They're made of DNA. And DNA is all your genetic information that's responsible for your traits. So we want to make sure there's no mistakes. Those are called mutations. Now, sometimes there is, and they don't get, they don't get checked over very well, and that could be a bad thing for the cell. The cell won't be able to operate correctly if the mutation is a bad one. Some mutations aren't really that big of a deal, but some of them are. But again, this is the phase here. I call it the proofreading phase. It's kind of where all that genetic information gets proofread to make sure that it's okay before we move into the next stage of the cycle. All right, so this is kind of where we're at. I mean, we've started here, a cell with 46. We've gone through all three steps of interphase, and now we have a cell that has 92 chromatids in it. Okay, and now we're ready to move on to the next stage. Now, at this point in the video, you might want to pause it, take a break. Okay, and then when you come back, we're going to continue with the cell cycle. Okay, so now we're going to move on to this area here. Okay, this is called mitosis, and again, you've probably heard of this before. Maybe you did a little bit of this in middle school. Mitosis is basically the division of the nucleus. So I want us to take a look here at this picture down here, right? Just kind of big picture, right? Here we have a cell that has a nucleus in it, okay? And let's just looking at the picture, it's one nucleus. Well, at the end of mitosis, you're still going to have one cell, even though it kind of looks like it's becoming two, but it's still one cell. But there's two nuclei. Okay. So what I want us to understand about mitosis is, how do we go from this, a cell with one nucleus, to this, a cell with two? Now, to understand it, we have to go through four different stages of mitosis. You've heard of these before, probably: prophase, metaphase, anaphase, telophase. I think of it as PMAT. That helps me remember the order. All right, prophase, metaphase, anaphase, telophase. So let's go through each individual step so we can kind of understand what's happening as we go from this to this. All right, so prophase. So here's our cell. Okay, this is the same cell that went through interphase, right? Went through the G1 and the S and the G2, and now we've got our cell. So a couple things happen here. Number one, the nuclear envelope. This is the membrane that's around the nucleus. It starts to break down. So basically, the nucleus starts to disappear. Another thing that happens is there's these two little structures. Well, I guess there's four of them. There's two of them here, and there's two of them here. These are called centrioles. Now, you'll know more about what they do in a minute, but those two centrioles start to migrate to two different sides of the cell. We call those poles, like opposite sides of the cell. Another thing is there's a structure called the spindle fiber. It's actually a collection of different fibers or different strands in the cell. Again, you'll see what they do in a minute, but these spindle fibers start to form during prophase. Okay. I still want us to remember that during prophase, we still have 92 chromatids. 4 number ones, 4 number twos, 4 number threes, 4 number sevens that are inside of this nucleus. Okay, remember that. Now we're going to go into metaphase. If you kind of look at the cell, you probably can describe to me what's happening. I take a short break here before I go into the details, but just ask yourself, what has happened from this picture to this picture? Okay, what's going on here? Well, one of the big things that's happened is those chromatids. I probably shouldn't use the word chromosomes here, but anyway, they've moved or they've migrated to the middle of the cell. We call that the equator of the cell. Now, you'll notice these spindles have now attached themselves to the centromere. Remember the rubber band that holds the two sticks together of each chromatid. Okay, that's a big thing. We still have 92 chromatids at this point. Okay, they're all just lined up at the equator. Now we're going to go into anaphase. Now again, if you look at the picture, you'll probably understand what's happening here, right? I mean, it just kind of looks like those chromatids are being pulled apart. The spindle fiber is pulling them apart. And once that occurs, then the spindle starts to break down a little bit. Once it's done its job, those spindle fibers start to break down. But once those chromatids are pulled apart from each other, then we can call them chromosomes again. So we're kind of changing our vocab up a little bit again here. So now we call them chromosomes again. But at this point, we still have these 92 chromatids. They're just kind of being pulled to either side of the cell. So we still have 46 going one way, 46 going the other way. And then we have our telophase, which is kind of the last phase of mitosis. So you can kind of, again, look and see what's happening here. Now, one thing I want you to understand about this picture is this is still technically one cell, but now you can see that we have the nuclear envelope starting to reform here. It's starting to reform here. So now we're getting those two nuclei that are beginning to kind of reshape themselves. But at this point, again, we've got 46 chromosomes up here. We have 46 chromosomes down here. So we still have one cell just with two sort of distinct nuclei. Now, the very last stage of the cell cycle is cytokinesis. Now, the term cyto, right, means cell. Kinesis means to split. So I'm showing you these two different animations here because I want you to kind of understand that there is a little bit of a difference between how an animal cell um, separates, those two cells separate, versus how a plant cell does it. Okay, let's first take a look at the upper picture here, the upper animation. So here's your prophase. Okay, centrioles are moving. Here's your metaphase. There's your anaphase. And then here's your telophase. But then look what happens. Then we get that cell to kind of cleave itself in half, kind of cut in half. We call that a furrow. A furrow is when the cell kind of pinches itself and eventually splits. But we don't get two definite cells until it completely separates. Okay. So this is what happens in an animal cell. Now down at the bottom, it's a little bit different. This is a plant cell. So instead of that furrow forming, instead what we get is basically a whole new cell wall. We call it a cell plate that kind of forms in the middle that sort of separates the two cells out. So there's a little bit of a difference there between plant and animal cells, but nothing really major. Okay. All right, guys, there's a lot that I threw at you today. Okay. Again, our job is to be able to describe, what happens in interphase, what happens in mitosis, what happens in cytokinesis. And what I might do if I was you is I would break it down by each one. Study interphase by itself. Know what happens in G1, S, G2. What does the cell look like at the end of interphase? All right, then go on to mitosis. What's happening in prophase, metaphase, anaphase, and telophase? What does the cell look like at the beginning of mitosis? What's it going to look like at the end? Think about chromosome number as well. And then finally, cytokinesis. What happens during cytokinesis? How does it differ between plant and animal cells? Those are all good things to know. All right, guys, I will let you go. If you have any questions, feel free to email me or just chat with me on GoGuardian. I would rewatch this video maybe once or twice before you take the next quiz, but really try to study this material so that you understand it. All right, guys, have a good day.